we'll hand straight on to Raja, who is tantalizingly titled talk, A Quick Trip Through Risk Prevalence and Everything Else in Between. So, Raja, over to you.
Thank you. Can you hear me? I'm hoping so. Right. Okay, let's share this first. And I'm not sure if I'll see anybody once it's shared, so I'm hoping that it will work and it will all... Right, so I'm presuming you can all see slide share and all that kind of stuff now. I can't see anybody, so when Greg pops up on the screen, I can just carry on talking because I can't see him at all, so it's all fine. He might have to shout at me. Yes, so between myself and Cassie, who's coming next, we decided to share out the workload. So rather than us focus, so I'm going to focus on the first part, which is more to do with the background, some of the features that go with it, the prevalence and those bits. And Cassie's going to focus on the neuropsychological and the cognitive bits a bit later. Maybe a slight overlap, but not much. So hopefully between the two of us, we'll cover the background and the basics of it. And it is a rapid run through. We can't spend 5 minutes on each slide because of the fact, and you will get the handouts from this in order to do that. So you have to see this as an introduction and as a starting point, not an end point. You won't go away knowing everything from these because, in fact, you can't. It's simple as that. I will highlight my new book, which is literally at the end, which is... Hello?
Raja, just we can hear you, but we can't see your slides. Are you sure your slide's sharing?
Hold on. May not be. Let me...
I'll tell you when I can
share. Can you see that now?
Yes, champion. Thank you.
Good. Thank you. Right. All right. So you could have heard me. So all you've seen so far is my intro slide. So you've not missed anything. So that's a good thing. Yes. So effectively what we've got is that we're going to run through it. Now, you've already heard from Joe about the diagnostic criteria. And what we've ended up with is from the Institute of Medicine, which is in 1996, they met together to try and define what was a small, narrow band of people into a broader understanding that this is a broader presentation. Since then, Susan Astley and Sterling Crowing developed the 4-digit code. The Canadian guidance came out, and you've had different groupings. But we've ended up with the Scottish government really investing and actually pushing this. They put together a committee, the Sign Guidance Committee, which is equivalent of NICE, that developed a review, and they came up with the conclusion that we would be FASD with and without dysmorphic features. And as NICE has adopted that lock, stock and barrel as their view going forward, that's where we're going to end up. So the diagnostic criteria we'll be using in England will be FASD with and without dysmorphic features. Now, part of the consensus and the arguments that you will hear in the press is that we all disagree. Well, I can tell you, being on this international research group with all the people who've written all the different criteria, is we don't disagree on the fundamentals of FASD. What we disagree on is where the cutoff should be. And this international group has been pulled together to try and look at that and to develop some research criteria around it. And that work is ongoing at the moment. But this group involves everybody who wrote all of those previous panels and the research criteria. So hopefully that will, when it's finished, the work will help bring this together. Because if you look at where around the world that these things have occurred, you can see immediately that there is a whole range of places in the world that this has been done. It's not just one person. If it's just Raj is making all this up as he goes along, you could argue maybe I'm slightly out there or whatever. But actually, if you look at the work that we've done in our clinic compared to places around the world, this is a comparison with a Canadian study. The profile on the brief, you can see the shape is very similar. Ours is slightly higher because we have a more clinic-based population, and that their study was based on a community sample. But the profile is the same. And if you're getting the same profile and the same principles in different parts of the world, we're dealing with the same thing. And so what it comes down to is, do you want to call it FASD? Do you want to call it something else? But the same thing is being addressed. And that's the first fundamental point. When we think about alcohol, well, we have to think, well, what actually is it doing? Well, what we know is that all of us are created by our genetics, and they influence the amino acids that create proteins that leads to us. When those genes don't work properly, amino acids are not properly coded, or there's something about the process that goes wrong, and it leads to malformations. Well, when we're dealing with alcohol, it's more complex because it works in multiple ways, but the direct and indirect effects of it, the direct effect is when alcohol is in the system, it is interfering with the process of those things being produced. And so whether it's killing off cells, whether it's interfering with neurochemicals which draw cells to different places, as we'll see later, it's interfering with the process. So it has a direct effect, but it also can have an indirect effect. And if you can see these dots here, the white and the black dots, they're a graphical representation of how you can change how genes are expressed. And this is what we call epigenetics. And so by adding methyl groups to this coding bit just before the color gene, you get different coat colors for the different mice. And it shows that there are also indirect as well as direct effects. So it's a complex interaction that this has, rather than saying this is very straightforward and understanding. Why there are variability and differences between people exposed at different levels is because there is a complexity to individual behavior, which is to do with environmental exposure, genetics, and a work. So it's not a straightforward linear A leads to B, and that's part of the problem. Now, the next question, therefore, is, okay, we know it's complicated, so how many people are drinking? And this was a study that came out in 2017. It's still the best systematic review that's been done, and it identified that we are the fourth highest consumer of alcohol in pregnancy, with 41% of women drinking at some point during pregnancy in recorded literature. Now, this is a scientific study that has inclusion and exclusion criteria, and so therefore it was only taking ones that have done robust research. But either way, it identifies that this is a very high problem. A study that was done in Leeds looked at dietary and lifestyle advice throughout pregnancy, and alcohol was one of the things they looked at. And if you look at the area in the red circle, what you can see that around 27% of women had drunk more than recommended levels throughout pregnancy, and around 48% had drunk at some point during pregnancy. Now, we know that people underestimate, so it's highlighting that there is a risk that is ongoing, and that at that point, at least when this study was done, that there was a consumption and an ongoing effect. So how common is it? Well, we now have studies in the UK. When I used to do this up to a year ago, we used to say there's no studies that have been completed in the UK. Now, there are two studies that have been done in the UK trying to get an estimate of prevalence. The first one was done by Cheryl Maguire as part of her PhD. I was able to help her with part of this. And what she did is take the ALSBAC database, which has 15,000 people, and apply standards. It's more a screening assessment rather than a direct assessment, because it's indirectly done. It's not done on the people who were looking for that. And what you can see here from this is everybody had alcohol exposure, and everybody had to have the neurological deficits in keeping with the diagnostic criteria, but around 2% had full facial characteristics. So only 2% of them, roughly, would have met full fetal alcohol syndrome. So anybody who's looking for just that, this is a rare presentation in the UK. If you're looking at partial phenotype, it's closer to 10% that Joe was talking about. But what you can see is that actually there's a lot more people who are hidden where the characteristics are not necessarily there. They came out with an imputation between about 6 to 7% for the full one, but for any effect of alcohol, because anything up to 17%. And so the rates there were potentially quite wide and quite high. We've literally just had accepted our paper on prevalence, which should be coming out very shortly. But Penny was the lead for it. I was a co-applicant on this paper, and we did a study where we looked at it. And what we found for those people where we confirmed and where the alcohol exposure was most likely, it was 1.8%. We had another group of people where it was probable there was information missing, but would have taken it close to 4%. And so it's around 2 to 4% of the population are likely to have FASD based on that. Now, there are limitations that it was a small study. It was only done in Greater Manchester, but it was the first active ascertainment study of its kind, thankfully commissioned by some of the people who you're going to hear from later on. But it gives us some insight into what we're dealing with and how far we need to go. We now want to replicate this on a much bigger sample so we can get a proper estimate. If you look in certain subgroups, for example, the looked after children's group, you can start to see again the rates are far higher. These days, you're not taken into care unless there is a difficulty with families. Very rare do you get a healthy child taken away from a single mum, which was fairly common. Well, it wasn't common, but it existed a lot more in the past. These days, that happens much less. So for people being taken into care, there are often issues. And drug and alcohol is one of those things that will often be part of that presentation. Two studies done in the UK, both are more indirect studies where they've estimated groups, and a third in the study in near Middleton Keynes in a looked after children's population. And again, a study by Jez Gregory in Cambridgeshire identified a third of her population that she had in the looked after children's group, where she audited against diagnostic criteria all the children she'd had for a year as a looked after children's paediatrician. A third would meet criteria for FASD, and 3/4 of those who adopted potentially met criteria. So when we're looking at this, we have to look at this as a condition that is not just the positives ruling in bit, we have to rule things out. And there's a whole range of things that we need to consider when we think about, because life is not about one thing or another. It's not just about alcohol. It's not just about your genetics. It's not just about postnatal life. What our clinic does and what more of the specialist services do is try and pull apart all these different facets to get to the point and say, well, actually, do you know what? This is what we think is left over, and therefore, in quoting great Sherlock Holmes, if everything else is been shown to be ruled out, then what's left over must be the truth. And that's kind of what you're trying to get at, is to try and rule out as much as possible, so you're left with a conclusion and an understanding. And so what we tend to say is that genetic disorders are the most common cause of neurodevelopmental. Do a microarray, rule those out. We're not looking for positive results. We're looking for results which are negative. Prematurity, we know when people are born, after 34 weeks, generally there is no ongoing care, and only about 10% of premature kids go on to have neurodevelopmental issues. Postnatal neglect we're going to talk about, but then medication, smoking, they have different effects to alcohol, so we can look and identify that. And then there's postnatal drugs. If we look at postnatal drugs, a review done in 2013 showed that of all of the conditions that affect, alcohol has the strongest short-term and long-term effect. The way that you look at this is A effect is a mild one, is in terms of a correlation of about 0.2 to 0.3. This, we're talking about strong effect, is a correlation of about 0.6, 0.7 or above. And so there is a strong relationship between these different things. And what we're starting to understand is it's not just here marijuana, it's the type of marijuana you have compared to skunk or other things. So the amount of THC has more of an effect. But alcohol overall, from when this was reviewed, has the strongest ongoing effect of all of them, which is surprising to a lot of people when I talk about this, because they think things like heroin and opiates are going to be stronger. But you can see the effect is far less, especially in the long term, in those kind of situations. One of the things that always struck me, especially when I first got into this, was that relationship between etiology, environment, biology, and how it fits. And it felt to me that you had to be in one camp or the other, and you weren't allowed to think about the relationship between them. Because the reality of it is that what we have is that these two things interact, and you're looking at the symptoms and you're trying to pull apart which bit of this is the biology and which bit of this is the environmental issue. And what we came to is, I coined it as a phrase as a psychiatric onion, and my wider neurodevelopmental team has now adopted this and telling everybody about my onions, which sounds a bit odd, but it's a way of understanding that you have to look through the lens of the neurodevelopmental presentation first, because that is what leads to the vulnerability to the world around them. And then you can start to think how the environment influences all of this. Now, because so much of the world is understanding in terms of attachment and other factors, like I wanted to go back and understand how these things interrelate. And so if we look at attachment in terms of basic principles, a typically developing child will go through a period where, 0 to 6 weeks, they kind of just are there, they eat, sleep, they poo, and they cuddle you. By this later on, they start to make indiscriminate attachments, and that 6 to 7 months is an important time frame, because we're going to look at the neglect aspect in a second. But then by those 6, 7 months, you start to make an individual attachment going to multiple attachments. That's how standard attachment works. And it's a biological principle in terms of safety of a child as they're developing and making that link towards the parent who is going to look after them. When that doesn't work properly, you start to get attachment difficulties and those reactive adjustment problems which occur when parenting styles get involved. But what happens then is it's all blamed parents' faults, child's response. It doesn't say how does the child influence this presentation as well. What does the biology of the brain innately lead to a difference? And you have to understand that in terms of the situation. And what we end up doing is, without understanding the big picture, is we start to blame. And we've seen that so much. And this is a piece of work that I did in 2013. It's been replicated since then. in other studies, where what you hear is families have to fight for things, and they're often misunderstood and blamed, and they're often taken away, and people are saying it's your fault for being a bad parent, not understanding that there may be dialogical issues that we need to understand also in how the child processes the world around them, how the adult then goes on to have expectations they can't manage, and that as much needs to be understood, because what we talk about is a complex trauma model which actually envelops the individuals. We did a study looking at this, and what we found is that people with prenatal alcohol are more likely to have a disorganized attachment style irrespective of the parenting, which is implying that the brain-related issues have an impact on that. Now, there are very few studies that had been taken place at that time. So we needed to try and understand, well, what does a wider neurodevelopmental world show? And what you've got is a whole load of studies that have looked at autism as an example, which is a social communication deficit. And actually, social communication problems are a core problem in people with FASD. The level of impact varies, but there's a difficulty for many. And what you can start to see is that insecure attachment patterns were far more common. And in this study by Rutgers in 2007, they found that parenting didn't influence the attachment behavior in the autistic group. Now, since then, what has been started to identify is that there may be some differences in the brain pathology between groups in the FASD group, but in the wider neurodevelopmental world, that postnatal neglect wasn't causing the level of neurological deficit, but having a neurological deficit or a neurodevelopmental problem increased your vulnerability to experiencing life problems. And that's partly what we started to understand and see. And we wanted to try and pull that apart. And so we did a piece of research and we looked at it. Now, if we take that back, the big study which shows environment and neglect can cause neurodevelopmental issue was the Romanian study. And that was suggested that actually, if you remained in care after 6 months, and the level of neglect there was 60 children to one adult, so severe neglect, extreme neglect, that you started to have these neurodevelopmental outcomes. Studies in, for example, in America, Tizard and Hodges, for example, where the ratio was 1:4, you didn't see so many things. So we wanted to try and understand, well, what are we getting? How do we delineate that? PhD student Alan did a review and basically found that there was a whole group of people that had done stuff and it was heterogeneous in terms of their finding, is that vulnerabilities were more likely to be there, but there was no consistent pattern in terms of what the different people, so drawing conclusions was very difficult. So we wanted to start again. In our clinic, what we were seeing is a group of people had both prenatal alcohol and neglect. Some people taking to care straight away, others not. What we didn't have was just the neglect group without any prenatal alcohol, as you wouldn't expect in a specialist FASD group. Because what you need to do is there start to compare and see other differences. And when we looked at this, you could see when we looked at neurodevelopmental outcomes, there was no difference whether you had no neglect at all, up to 6 months, or protracted neglect. There was just no difference between any of these groups. And that starts to get into why the differences were seen. So we drew the conclusions that neurodevelopmental disorders were independent. The brain parts were separate to neglect, and neglect didn't make those neurodevelopmental presentations any worse. So then you start to ask yourself, well, what does neglect do on top of that, and how does it differ? And there was a study where you need to compare these two groups. And there was a study that had already been done in America, and Alan's work had started to compare some of that. And what you could see is that the neurodevelopmental group with PAE as well had far more of the neurodevelopmental presentations, whereas the psychological stuff goes back to my onion of the brain, environment, trauma, the experience later on. And that's what you're seeing, is that the neurological stuff seems to be prenatal or genetic or biological, and that changes their vulnerability to the world and how they react to the world around them. And that's partly what we're starting to see. And this is unpublished research. It's not shared with anybody else. You won't have it in your pack. But what Larry Bird shared with me, we did a quick review of his database when he had it, and he showed exactly the same thing, is that the people with just adverse child experiences had much less rates of neurodevelopmental problem compared to those where you had FASD as part of it as well. So confirming our findings in a different setting. But what it does do is it leads to increased vulnerability. And this is a study that Alan did as part of his PhD. And what you can see very clearly is that the rates of neglect and the outcomes on the Adverse Child Experiences questionnaire were far higher across the board in all aspects, apart from sexual abuse and physical abuse. But even there was still quite high. And so the experience of adverse effects is far greater. And when you start to break this down, all you can see is all the groups that have looked at this have identified recently, until the most recent one in 2021 by Tan et al., that adverse childhood experiences are far more common and lead to a psychological trauma on top. So early identification, early diagnosis, changing the life trajectory can prevent those extra things happening. Even if you can't change some of the neurodevelopmental presentations, you can learn how to live with those, how to adapt and prevent the subsequent trauma. So it's really important that diagnosis occurs early and that we stop all these other things happening. Because actually, if the brain doesn't work properly, then the roots of the tree aren't there and the tree doesn't grow. And attachment is partly linked to neural functioning. And if the social communication centers of the brain, those things that are linked to attachment, are already damaged prenatally, which for whatever cause is going to have an impact. And that's what we're continuing to see. So it's a way of thinking about, instead of the child having a brain-related disorder, missing it, having it then leading to poor understanding, the child is blamed for being disobedient, and we label as conduct disorder. What should have happened is the brain is understood, communication is therefore adapted, people around adapt to the individual, and it leads to a better quality of life, and the disability that is understood is supported, and therefore you don't get the secondary labels, the secondary problems. And it's a way of thinking different about it within that complex trauma. We need to understand the experience of the individual from their perspective, looking at that, not our perspective, not the lens of a typically functioning person, but what is the lens of that individual to take it through? How does their brain process information, and how does that work in terms of those other factors? Because otherwise what we're doing is perpetuating that trauma. So how does it present to us? Well, 4 broad ways: growth, behavior, learning, and physical problems are the common. Growth to physical health problems, behavioral issues to people like myself as psychiatrists, learning issues often at school, and physical problems to a whole range of people. I was involved in a study which we published in The Lancet in 2016, and we looked at the criteria and we identified there are 428 different comorbid conditions across ICD-10, from conduct disorder, language disorder, intellectual impairments, but a whole range of physical health issues by which they present. They don't present very often saying, this is FASD. FASD is the cause, the etiological relationship between these, but they present with the outcomes. And that's what you have to look back and say, well, actually, what is underlying this? Because the different causes have different treatments, and it's important to understand that. The other thing to understand is it doesn't necessarily just about how it presents, but when it presents, because the gap becomes bigger as you get further on, because there's a developmental divergence as things progress. And this is another unpublished study. This is from a colleague in Canada, which you won't be able to have access to. But what it shows is, when you just look at the top line, which is highlighted, if you tested just IQ up to the age of 12, only about a third would meet criteria. By the time they became 18, nearly 2/3 were meeting criteria. It's to do with the gap getting bigger and the trajectories being different. That doesn't mean their IQ is deteriorating. It means the normal, typical population where we do these tests and normalize them and get those standard norms, the gap is getting bigger from that. And so that is then expectation is placed upon people when they're assessed in primary school. Doesn't necessarily rate. So reassessment is important. Understanding where their function is at different stages of their lives become an important factor. So what do you do to diagnose it? And I'm not going to talk at great length about this because I'm going to run out of time if I do. But effectively, as I've said, there are 4 broad areas that we look at: face, growth, neurological areas, and alcohol exposure. The difficulty with alcohol exposure is not written down well. If any of you and the social workers in the audience, one of the things I would plead with you is just note down the alcohol. Any pediatricians, note down the alcohol. One of the second nice guidance is because one of the biggest reasons why people don't get a diagnosis is it hasn't been written down, and people are therefore reluctant to give a diagnosis. Now, if someone's lifestyle is very much chaotic alcohol drug use before, chaotic alcohol and drug use after, it's highly unlikely they stop absolutely at the point of conception through to the end point of the pregnancy. It may have happened. And so, but the more accurate detail we have, people like myself are able to give accurate diagnoses. There's another paper that just come out from Australia, which basically has suggested that teenage drinking patterns are very predictive of later pregnancy type drinking patterns. It doesn't change significantly through that period of time. What we see now, however, is when I first started this 20 years ago, 2/3 of women drank during pregnancy. Now, 2/3 of women to 3/4 will stop once they realize they're pregnant, not before. And patterns of behavior change at conception, or if they're planning pregnancy, not at the point of of the test, sorry, not at the point of conception. So that can vary as well. So it's asking accurate questions. And you ask questions as part of a lifestyle history, not as part of a prerogative. Do you drink? And that's terrible, isn't it? You don't do that. And it's where there are ways of looking at all of this. So what is the diagnostic process? And what's interesting here is this comes from 1984, and it hasn't changed significantly since then. So the short palpebral fissures, indistinct philtrum and upper lip are the core features we look at. Now, there are two other features here. And why are they not used then? Well, flat midface, because you couldn't measure it back then. With 3D imaging coming through, we're starting to be able to manage it. Head circumference is an additive feature in terms of the cognitive, but it's not a discriminating. Not having it does not exclude it. And that's what people used to say. So that's an important factor. And they get a whole range of associated features as well. The other thing that we know from a very early on is there's a timing effect, is that when you drink during pregnancy at certain points in time, you're at risk. So here in a mouse model, on day 7, you have the features of FAS. Day later, you don't. And that's showing that there is a timing effect which relates to the palate and lip being created in a 2-week window, roughly between week 4 and 8. And so we're defining an entire syndrome with, if you just look for just the physical features of FAS, in a 2-week period during a 40-week pregnancy, whereas the brain starts developing at day 18 and continues to develop throughout, with different things happening at different stages. And Cassie's going to talk more about the cognitive bits later. What we've moved on to now is 3D imaging, and it far better and far more accurately measures things. So this has 20,000 landmarks, and we're working with Oxford University to try and embed this into a clinical practice. And we've been working with them for a while as to how to develop this. And we've got a couple of papers, hopefully, we're working on to try and get out around that. But it's embedding this into clinical practice. Now, maybe this is a specialist center kind of approach, and we use the 2D at other levels, but that's to be discussed later on. Growth. Again, it comes down to when do you assess it. So for this person, if they came to clinic at this point and we didn't have any records, they wouldn't have met criteria. Whereas when they're early weeks and early months, they would. And so having that red book, having that information is really important. Documentation is so crucial to those of us who are seeing people 6, 7, 8, 10 adult life down the road. And what you get later on is not necessarily what we see at the start. Cognitive domain, I'm not going to deal with. I'm going to let Kes deal with that next. And then what we're saying, well, what's next? Well, Joe's already mentioned the quality standards. I was the expert member on the panel, sort of running the National Clinic. And what we can see is these are going to hopefully come out soon. It's been delayed by a long period by COVID, so it was cancelled. They should have been out last year. But at the moment, we've had our second meeting, and we're just waiting for ratification, and then hopefully they will come out some point this year. In terms of that, there were legal challenges against some of this, so it's being taken a bit more cautiously because of the nature of capacity, consent, women's rights, and having the ability to choose. And what I would say is we're very much in favor of giving information to women, letting them make their own decisions, but let's give them the correct information about what we know and what we don't know, and let people make those choices. So that's my clinical team, and that's us done. And that's my book that came out 2 weeks ago, and it's designed for clinicians as a starting point. So if anybody's interested, it is currently available if anybody wants it. I don't get any fees from it. It goes straight to Springer, but it's hopefully it's a good starting point. And that's me done.